Dr. Andrews leads the scientific research and development function of Sierra Sciences. His team is now pursuing direct molecular approaches to temporarily activate telomerase expression and is performing discovery efforts. While director of the molecular biology at Geron Corporation, he was one of the principal discoverers of the telomerase gene. I'm proud to introduce Dr. William H. Andrews.
Groucho Marx once said, "I intend to live forever or die trying." He was trying to be funny when he said this, and he got a lot of laughs. But I don't believe the people in this room think this is very funny, because we're here because we're all trying to live forever. I know I am. And we are going to try our best to do that. And so what I'm going to be talking today isn't really how to live forever. I'm going to be talking about how to try to live forever. I'm going to be talking about research science that's underway now that's going to allow us or increases our chances of succeeding at this. And so pretty much what I'm going to be talking about today is not what you can buy today. In some cases that's not true. But I'm going to be talking about what's in the future, what we have in the future to look forward to. So how to try to live forever, first do the obvious things. Eat healthy, live healthy, exercise, listen to everything your doctor says. But this isn't enough. This isn't going to allow us to do things like run 7-minute miles when we're 130 years old. And that's what we want to do. And by the way, we're not just talking about living longer, we're talking about living healthy longer. We want to be very healthy when we're in our hundreds. Now, there's been a lot of things that have allowed us to extend our lifespan tremendously, especially just in the last 100 years. In 1900, the average lifespan was only 47.3 years. And since then, we've extended that up to 78 years through a lot of things, including better sewage systems, vaccines, refrigeration, exercise. The other column has coronary artery bypass surgery, chemotherapy. All these things add up to give us a tremendous extension of life. But none of these things are going to cure aging. We still aren't going to see anybody, or we aren't going to be running 7-minute miles when we're 130 years old. And this is because there's a theoretical maximum lifespan that a lot of scientists have calculated to be 125 years. Apparently nobody can live beyond 125 years. There's been at least 3 studies done. References are here. The most recent one was just a few months ago, a mathematical model looking at how much older people have been getting over the last 100 years, and still they see this threshold. We can't seem to pass this threshold of 125 years, and they don't know why. Most of you have probably never heard of a telomere, and so I'm going to give you a basic background on what this is first. And to do this, I need to zoom in on a person. And when we zoom in on a person, we see that a person is made up of 100 trillion cells. And all those theories I was talking about before on why we age, they all relate to the aging of our cells. We age because our cells age. If we could find a way to prevent aging in our cells, that would probably cure our own aging. If we zoom in even further, we see that every cell contains a nucleus. And inside these nuclei, there are chromosomes. And these chromosomes are where all the genes exist to give us our blue eyes or blonde hair, make some of us short and make some of us smart. And so I want to zoom in even further now and explain that a chromosome is made up of a DNA strand that extends from one end all the way to the other. And the DNA is approximately 10 million bases long. It's made up of units called bases, and it's pretty much coiled up like a slinky all the way through this chromosome. The key of my talk today is the very tips of the chromosomes. These are what are called the telomeres. And if we unravel this slinky in the telomere, we can see that a telomere is about 15,000 bases long. At least it is when you're first conceived. As soon as cells start dividing, your telomeres start getting shorter. And by the time you have enough cell division to become a newborn baby, the telomeres have shortened already down to 10,000 bases. They've gone from 15,000 bases to 10,000 bases. As cells start to divide further, for us to become adults, our telomeres shorten even further. And when they get as short as 5,000 bases, we die of old age. Telomeres cannot get shorter than 5,000 bases. Inside of petri dishes, when we grow cells in petri dishes, the cells absolutely stop growing and die of old age at 5,000 bases. Let me repeat this. We are born at 15,000 bases. We are conceived at 15,000 bases. We're born at 10,000 bases. We die of old age at 5,000 bases. And there's absolutely nothing we can do about this. This telomere shortening is going to occur no matter how well you exercise, no matter how well you eat, no matter how much you do everything your doctor tells you to do. You cannot stop this telomere shortening. Now, this is the only clock of aging that anybody has ever discovered in humans. And it actually fairly well explains this 125-year theoretical maximum we have, because these telomeres shorten approximately 1,000 bases per year. So this is a new word. A lot of you now know what this word means, but I predict that in the next 5 to 10 years, this is going to become a household word. Everybody's going to want to know how long their telomeres are. I believe this is going to be the biggest and best biomarker of aging and overall health that we've ever seen. So the best example of where telomeres play a role in aging is the fact that there are some people who are born with already short telomeres. This disease, called progeria, affects these kids. And this disease is called the disease of short telomeres. These kids have a maximum life expectancy of about 20 years, and they suffer from all the same age-related ailments that normal old people do. If we could find a way to stop telomere shortening or to slow it down, this could be a cure for this disease. That's pretty much going to be what I'm going to be talking about today, is finding ways to stop this telomere shortening. Now, these kids aren't the only ones that suffer from telomere shortening. We all do. Telomere shortening has been implicated in almost all aspects of normal aging. And what I'd like to do is I'd like to just give a few examples, and I'll start off with the number 1 and 2 causes of death. Cancer. When our telomeres get short, our chromosomes become unstable, and the rearrangements of our chromosomes cause mutations, and these can induce the expression of oncogenes and de-repressed tumor suppressor genes, increasing our risk of getting cancer. In addition, when the telomeres in our immune cells get short, our immune cells lose the ability to fight the cancers. So it's two things happening here. Short telomeres increase our risk of getting cancer and decrease our ability to fight the cancer. Atherosclerosis. When you look at the cells lining your blood vessels, the endothelial cells and the smooth muscle cells, you'll find that if you look at the telomeres in the cells surrounding plaque, you'll find those telomeres are shorter than the telomeres in other parts of the blood vessels. And this is because we think that the cells have gotten older through telomere shortening, and as a result, they are less capable of preventing plaque. So if we could find a way to prevent that telomere shortening, this could be a possible way of preventing plaque and even fighting plaque. There's a lot of disease, lack of time. I'm not going to go into all of them, but people are welcome to talk to me throughout this conference. I can tell you the basics of telomere biology in all of these, but they include Alzheimer's, osteoarthritis, osteoporosis, just general skin aging. They include macular degeneration, which is the #1 cause of blindness in the elderly, liver cirrhosis, muscular dystrophy, AIDS. Now, you all remember back in the early 80s when AIDS epidemic first came around, doctors were saying that they don't understand why their patients don't have any T cells. But we now know that the immune cells, the T cells in the immune system, put up such a fight against the AIDS virus, and the fight was unsuccessful, but because of this fight there was lots of cell division, and the telomeres got very short in these immune cells, and the immune system died of old age. And that's why the AIDS patients don't have T cells. Now, if we could find a way to prevent the telomere shortening in these immune cells, the immune cells would stay around. They probably wouldn't be successful at fighting the disease, but the person might be able to live a normal life. General immunity. Stem cells. Every time I go to a stem cell conference, doctors are asking me, have we come up with a drug to prevent telomere shortening yet? And I always have to say, not yet. But the reason why they're asking is because when you take stem cells out of a person, you have to expand them in culture. You have to get more numbers of them. You have to let them divide. Well, because of the cell division, the telomeres are getting shorter, and so the cells that they put back into a person end up actually being older than the cells they took out of a person. So the people doing stem cells need a drug that will prevent telomere shortening when they're doing stem cell therapy. There's a lot of diseases. Anything you can think about that involves cell division is going to have telomere shortening. And one thing I want to leave you with is that bad things happen when telomeres get short. There's nothing good about short telomeres. And there's also nothing bad about long telomeres. So it would be good if we could find a way to keep the tips of our chromosomes long. So now let me address the question of why do telomeres shorten? And to do this, I have to explain, when I was showing you the cell and it had the chromosomes in it, when that cell divides, all the chromosomes, the two new cells that get made out of this have to have all the same chromosomes that the original cell had. So the DNA has to duplicate. It has to replicate. Now, DNA replication is a lot like laying bricks on a brick wall. The bottom strand here can be considered the original strand, and then the new strand, the top layer is the new strand, and this has to be identical to the original strand. Remember, this DNA is about 100 million bases long, so it's a long, arduous process. And if there's any mutations that are made during this thing, or any mistakes, this can lead to mutations that can cause cancer, etc. So this is a pretty accurate procedure. But what we want to do is we want to look what's happening over at the very tips of the chromosome, okay, the telomeres. And so we have this brick layer, which, by the way, in a cell is called DNA polymerase I. Is moving along, replicating, and you're going to see there's a problem coming up here. And this is what actually happens inside of a cell. The cell is unable to replicate the very end of the chromosome. As a result, every time DNA or chromosomes are replicated, they are a little bit shorter. And as I said before, there's nothing we can do about this. But I want to point out that there was no degradation here. There was no enzymes that came along and chewed this away. This is purely due to a lack of an ability for the cell to replicate the very ends of the chromosome. It's more of a passive, not an active phenomenon. But now this DNA has to divide, or the cell has to divide, the chromosome needs to replicate again. The new chromosome is even shorter, and again and again. And there's absolutely nothing we can do about this, no matter how well you eat, no matter how much you exercise, no matter how much you do, everything your doctor tells you to do, you cannot stop this telomere shortening. I said you can't stop it, but there are things you can do to accelerate this telomere shortening. And this has to do with anything related to an unhealthy lifestyle. And that includes things like obesity, lack of exercise, psychological distress, and smoking. Things like those will create free radicals inside your cells that will come along, and the free radicals will clip your telomeres and make them shorter. Now, this kind of stuff you can control. You've probably seen press releases lately where people are saying that simple meditation can help reduce telomere shortening. Well, this is why. It's reducing the accelerated telomere shortening, but there's absolutely nothing that will work to actually reduce this basal level of telomere shortening here that I described first. So, how are we going to stop this? I've been saying that there's nothing you can do about this, but that's all I'm trying to do, is I'm trying to figure out a way to do this. And I've learned from a lesson from our reproductive cells. Our reproductive cells do not age. And this is important because if they did age, our children would be born as old as we are. But they don't age, and that's why our children are born young. So something inside our reproductive cells prevents them from aging. And so this was a key to try to find out how to make our other cells not age. Well, as it turns out, our reproductive cells do not have telomere shortening, and this is probably why they don't age. And this is because, we discovered this about 15 years ago, our reproductive cells contain an enzyme called telomerase. And this is a picture of a cartoon of telomerase. The green is the DNA, showing the very telomere tip of the DNA. And the factory-looking thing is the telomerase enzyme. And what it's doing there is it's lengthening the telomere. Now, so let's go back to this original model here of the bricklayer as an analogy for DNA replication. This bricklayer is still going to fall off in a reproductive cell. But like an angel, telomerase comes in and replaces that brick or fills in the missing DNA strand. So as a result, our telomeres don't get shorter. And if you think about it, it can't be getting shorter because you came from cells from your parents, they came from cells from your grandparents, et cetera, et cetera. Your children are going to come from cells from you. And if we had telomere shortening all the way through here, you would never, ever be alive right now because you wouldn't have any DNA left. There'd be so much telomere shortening. So our simple strategy in my labs is to take into account that first, somatic cells don't produce telomerase. That's the problem. But they do contain the gene for telomerase, so which means they have the ability to produce telomerase, they just don't. And they don't because the gene is shut off. And so our simple strategy, obviously, is let's figure out a way to turn it back on again. So inside the reproductive cell, telomerase is produced by the telomerase gene. And next to every gene, there's a regulatory element that's like a light switch that turns the gene on or off. In this case, in the reproductive cells, the gene is turned on. So in all of our other cells, a repressor comes along and binds to that regulatory element and shuts the gene off. And that's called a repressor. So what we want to do in my labs is we want to find a drug that will bind to that repressor, remove that repressor from the regulatory element, and allow telomerase to turn back on. If you decide you don't want telomerase to stay on forever, you can quit taking the drug and the repressor will go back and shut it off again. So that drug would be considered an anti-aging drug. This is the team working in my labs, where there's about 30 scientists. We have a 10,000 square foot research facility in Reno, Nevada. We've been in operation now for 10 years. And we have $2 million robots that are spending all their time just screening drugs after drugs after drugs. And you can see like a robotic arm right there that will carry like 96 different drugs at a time, carry from station to station, and analyze, have equipment on this robot that will analyze the weather, see whether telomerase turned on. The key is, I want everybody to realize there's a very significant effort going on here to try to cure aging. And I have to admit, one of the main reasons this is going on is because I want to cure my aging. I'm very, very motivated by that. So we've been in operation for 10 years, and as of just a week ago, we have screened 141,756 chemicals, and we have now found 32 hits. That is, we have found 32 of these green things that dislodge this repressor. That is 32 different chemicals that turn on the telomerase gene. But this could be the biggest thing that ever hit the planet. Telomerase induction might just actually change the way all of us live if it works the way we think it's going to work. Now, this is probably where I should end my talk, because this is where we're at right now. But I want to say that we don't know the answer to this question yet. Will telomerase induction cure aging? It's all theory right now. It's all hypothesis. And so we don't know the answer to this. But I want to say that there is a light at the end of this tunnel here. There is a lot of reason for a lot of hope. And that's because of proof of concept experiments that we have done over the years. Now, ideally, as I've been saying, we'd like to find a drug that would turn on the telomerase gene by just dislodging that repressor, but that drug doesn't exist yet. And so what we've done in the past is we've introduced the telomerase gene through a process that we call telomerizing. We take the gene, we insert it into the chromosome by carrying it on a virus, and then we do it in such a way that it's not regulated, and so that gene is producing telomerase. And so we've done that, and first let me say that when you take a normal cell, a normal skin cell, and grow it in culture, the x-axis here is the number of days in culture, and the y-axis is the number of cell divisions, you find that after a time the cells start slowing down and level off. This is called cell senescence, and that leveling off place is called the Hayflick limit. This has been true for all cells ever, but for the first time ever, when we telomerized human skin cells in 1997, we found that they did not level off. They kept on growing. Nobody had ever seen this before. These cells were the healthiest cells in the world, and they also had a tremendously decreased risk of getting mutations, including cancer. So we published this in 1998, made headline news all over the world, and it really opened the door for a lot more research on telomeres. But one big surprise in this study is that the telomeres didn't just stop shortening, as we expected. They actually got longer. So now this begged the question, did we actually make the cells younger? And this was not really our goal at the time, but let's take it. Now, the most exciting proof of concept is very recent data. This is proof of concept actually in humans. There actually is a commercially available nutraceutical that people can buy now for use in humans. And the company that's making this nutraceutical has provided us with some of it to do research on. And we have added it to cells, and we have found that it in fact induces telomerase expression. Not very much, but little is better than nothing. Even a little bit will at least decrease the rate of shortening. But now, it might even be working better than we thought. In collaboration with another lab, we took blood samples from people that are actually taking this nutraceutical now and looked at their telomeres. And even though we couldn't see the average telomere increasing, we could see that the shortest telomeres were increasing, which is telling us that this nutraceutical is inducing telomerase, and this telomerase is having an effect on the telomeres. When people that had been on the nutraceutical for more than a year were looked at, there were several different things that were seen. Some of these biomarkers a lot of you are familiar with, but CD28 minus T cells is one of the best biomarkers of aging, and they tend to increase when you get older. But the people taking this nutraceutical saw their T cells, CD28 negative T cells decrease. They also saw naive T cells increase, natural killer cells decrease, blood pressure decrease, skin elasticity increase. This is really exciting. This is a real proof of concept that telomerase induction is something that can really be used to potentially cure aging. Now, we still don't know the question, will telomerase induction cure aging? And that's because this new nutraceutical hasn't been around long enough to really answer the question. But things are looking good, and I'm really looking forward to seeing what happens. We also still don't know the answer to how long we will live if we induce telomerase. But I predict that people will definitely live beyond the 125-year theoretical maximum. So, in conclusion, I'd like to say that Groucho Marx may not have been joking. Maybe he really did want to try to live forever. I know that I do, and I know that my company is very motivated to doing this. Our logo is Cure Aging or Die Trying.
Only about a month after Bill delivered that talk for the Anti-Aging Medical Association of America, the 2009 Nobel Prize was awarded for how chromosomes are protected by telomeres and the enzyme telomerase. And then in 2010, this TV news report was released. And this really was the first proof of concept in a living animal.
And now, eternal youth. Is it in a cage around the corner? News tonight of a breakthrough for some pioneering mice. But we always wonder, what does a fountain of youth for rodents reveal for humans? Here's Sharon Alfonsi reporting.
I feel tremendous.
In the movie Cocoon, it's a swimming pool that turns back the clock for a group of senior citizens. But now, researchers have found a way not just to stop, but reverse the aging process. The key is something called a telomere. We all have them. They're the tips or caps of your chromosome, seen here in yellow. This is what it looks like in a young adult. But as you grow older, the telomeres become damaged and frayed, and as they stop working, we start aging, experiencing things like hearing and memory loss. Scientists took mice who were prematurely aged, added an enzyme, and essentially turned their telomeres back on. You can see it before the enzyme after.
Their brain function improved, their fertility was restored. It was a remarkable reversal of the aging process.
Look at this picture. The mouse on the right has bad skin, gray hair, and is balding. But the one on the left had its telomeres flipped back on.
And you could see that essentially you now have a dark coat color, that the hair is restored, that the coat has a nice healthy sheen to it.
Even more dramatic, the change in brain size. This is before. The mice had 75% of a normal brain, like a patient with severe Alzheimer's. But after the telomeres were reactivated, the brain returns to normal size. As for humans, while it is just one factor, scientists now say by looking at our blood cells and measuring those telomeres, you can get a better idea of how well you'll age. The longer the telomere, the better the chances for a more graceful aging.
And also in 2010, Dr. Bill Andrews started collaborating with John Anderson, looking for telomerase inducers or telomerase activators in plants that were suitable for human consumption. And the science is really very simple. Telomeres are our body clock. They shorten with normal cell division. Telomerase is an enzyme that causes the telomeres to lengthen. A gene to produce telomerase is present in every cell in your body. Telomerase production can be switched on by substances known as telomerase activators or telomerase inducers. And Dr. Bill Andrews and John Anderson found some natural products that are telomerase inducers, and they are in product B, or isogenesis. Bad things happen when the telomeres get short. Don't forget that. Now, product B was released in 2011, and it's been improved over the years and is now called isogenesis. And this interview, which was done with Bill Andrews in July 2019, was after he'd actually been taking the product for over 8 years. So not only did he develop it, but this is after he'd been on the product for 8 years.
I've done over 100 ultra marathons, not just 2. I didn't want you to describe me as some kind of wimp that only did 2. Yeah, I do them. I tend to like to do ultra marathons once a month at least. But you know, sometimes I'll go like 3 or 4 months without doing one. But I pretty much try to. I go through withdrawals if I don't do them often enough. I love the sport of ultra marathon running. My favorite distance is the 100-mile distance. I've done longer ones. I've done like 138 miles nonstop at 18,000 feet elevation in the Himalayas of northern India. I've done that. I've also done 135 miles through Death Valley in the middle of summer when the temperatures reached 130 degrees. And that was fun. But the thing that I'm shooting for most right now is world records for my age group. I'm just kicking butt with everybody in my age group and typically beating all the people, you know, 2 age groups below me. I'm 67, so I feel pretty awful if some 50-year-old beats me. But in 2.5 years, I turned 70, and well, I'm gonna just destroy all the world records. And a good friend of mine just broke all the records, and so he knows that I'm gonna go ahead and beat his records now. But that's 2.5 years from now.
Yeah, just share with us too, just a little bit, you know, about yourself. I mean, how did you get into, you know, telomeres, being obsessed with aging, and then the merge of meeting with John Anderson for the birth of isogenesis?
Well, my obsession with trying to cure aging began when I was 10 years old. And that was, my father planted the seed in me by telling me one day that, you know, because I was so interested in science and medicine, that I should, when I grow up, become a doctor and find a cure for aging. So I've been obsessed with it ever since, all through high school and college and stuff. Everybody knew that I was somebody that was focused on trying to cure aging. But I was really frustrated by the fact that none of the theories on aging made sense to me. And it didn't explain everything. I decided there had to be some kind of clock that's ticking inside of our cells that explains everything. And, you know, what was that clock? And then in the early 1990s, I was attending a seminar, and I was at an anti-aging conference, and one of the speakers talked about the fact that telomeres shorten as you age, and it's so accurate that he could measure the length of anybody's telomeres and tell them how old they are. more importantly, you can tell them how long it'll be before they die of old age. So I thought, wow, here's a clock. The length of your telomeres shortens at such a consistent rate that he can accurately measure a person's age. So I instantly approached him and asked him if anybody's figured out how to lengthen telomeres. And he said they've been working on it for years in collaboration with the people that later won the Nobel Prize. But he said after 2 years, they'd not made a scratch. They had no clue as to how to lengthen telomeres and what enzymes were involved. So the guy hired me to work at Geron Corporation to take that on after I told him I'll have it figured out in 3 months. And then 3 months and 17 days, me and my team discovered this enzyme called telomerase, and we put it into human cells and showed that it reversed aging in every way imaginable in human cells in a petri dish. That was God, in mid-1990s. And so, then a lot of groups have repeated those studies. They've done more than just human cells in petri dishes. They've done human skin grown on the back of a mouse. And they've actually done engineered mice. Mice don't, let me say right now, mice do not age like humans do. We're a very different animal. But scientists did engineer mice so they did age like humans through telomere shortening. And we were able to show that these mice got younger in every way imaginable when we lengthened the telomeres. It's the only thing that's ever done this. Everything you've ever heard of that touts itself as a cure for aging or something like that, all they do is provide you with a little more energy. They temporarily get rid of wrinkles, which always come back. But nothing actually really reverses aging until this. This is the first thing. It still hasn't been tested in humans, and that's only because the really potent stuff is just way astronomically expensive to be able to treat a whole human. But we're working on that. We've got now some clinical studies coming up where we will be treating humans to see if we can reverse aging. Now, these are $700,000 treatments. That's our cost to produce enough of it. In the meantime, people need to be doing everything they can to keep their telomeres as long as possible so that they are healthy when we actually do make this affordable for everybody. And so in the meantime, what they should be doing is isogenesis, because isogenesis is something that does induce telomerase to be produced in your cells. And that's all telomerase does is lengthen telomeres. Now, telomere lengthening and shortening is like a tug of war. And so you still have the shortening going on. In fact, in the absence of anything, all you got is shortening. Okay, so let's say going to your left to right or my left to right, we have shortening going on. And you can do things to accelerate that shortening by poor lifestyle choices, poor diet, things like that, smoking, stress, things like that. That can make it go really fast. But nothing in our body can actually make it go the other way, can pull it the other way. So when we do something like isogenesis and treat, so what we do is we find the cells start to produce this enzyme telomerase that can lengthen, but it doesn't win the tug of war. It still shortens. Okay, so nothing wins the tug of war. There's nothing available yet wins the tug of war. And so all it's really doing is kind of decreasing the rate of aging. Now, that said, we have found that when the telomeres get really short, they do start lengthening. Okay, so the critically short telomeres do get longer, and that's why everybody who's on isogenesis is seeing some age reversal things. Like even me, I've seen age spots disappear on my hands. I've seen my vision get better. I've seen my endurance get better, and it's only explainable by isogenesis.
It's really incredible, just a lot of the testimonies and even people, that they're in their 20s and they're 30 and just in recovery or, you know, other issues that they have been able to just see, you know, quality of life increases. So it's because we're always aging, no matter what age you are.
I'm trying to fix that. Yeah.
You're working all the time on it. So we appreciate that. So then how did it come about then, like your lab, Sierra Sciences, and John, how did that meeting?
Well, first of all, the company that I was working for when I first discovered telomeres, they decided suddenly to become a cancer research company. And I'd already been, have a big, strong history in cancer. In fact, my National Inventor of the Year award in 1997 was for my cancer research, not my aging research. So I was obsessed with aging, and when they decided to become a cancer company and stop all work on aging, I thought that was nonsense. And so I started Sierra Sciences to focus on the aging, and they gave me their blessing. Actually, we weren't competitors at all. So we were working for a long time, making a little bit of progress, not really happy with things. And then Peter Greenlaw, he one time called me up and was very interested in talking about telomeres. I ended up talking to him, and then he brought John Anderson to my company, and we talked for a long time. In fact, I was probably sitting right where I am right now during that meeting, talking to John Anderson on the other side of the table here. During that meeting, we outlined a plan where he would start sending us natural products to test, and we would see that if any of them caused telomerase to be reduced in human cells. We had already told him that, you know, we've already tested 2,000 different natural products, and not a single one of them worked. You know, but he said, you know, test mine, because most natural products that you buy on the market are prepared incorrectly. They've got solvents that destroy activity. They take them through heat treatments that destroy activities. He doesn't do any of that kind of stuff. He says that we could even test the same things we've already tested, prepared by other people, and he said we might find some that work. So first thing he does, so we signed the deal, we got it all organized. Me and the scientists were all excited, until he sent us 15 samples, okay? We thought, 15? We've already checked 2,000. There's no way 15 is gonna work. And it was a real struggle to even get the scientists to do it. But then, to our surprise, one of those 15 was a hit. And it was the first hit that we'd ever seen. So now we had tested 2,015, and in the last 15, one of them worked. We thought that was an extreme stroke of luck. And so John Anderson sent us another 15. And again, we found another hit. And that's when my scientists and I started really saying, okay, something's real here. In the long run, he sent us 10,000 different natural products over a long period of time. We tested every one of them. And we found 37 or 39, I can't remember, different plant extracts that induce telomerase. And what John did is he took the strongest ones and pulled them and put them together and called it product B, which later became isogenesis. We are still testing things, and that's why there's been a first generation, a second generation, a third generation, a fourth generation. And I'm not allowed to give too many details, but John Anderson has given me the okay to say that we are working on a fifth generation, okay? And we have found hits that are gonna be really enhancing the fifth generation when it comes out. But, you know, I would still encourage everybody to keep taking the fourth generation, because they definitely are gonna be a lot better off when we come out with the fifth generation if they've been taking the fourth generation during this layoff time. And I don't know when that's gonna be. A year from now, 2 years from now, that's totally a John Anderson isogenics decision, which I don't get any vote in at all. We have found that when the telomeres get really short, they do start lengthening. Okay, so the critically short telomeres do get longer, and that's why everybody who's on isogenesis is seeing some age reversal things. Like even me, I've seen age spots disappear on my hands. I've seen my vision get better. I've seen my endurance get better. And it's all explainable by isogenesis.
Bill, thank you so much. I know
Thank you. I enjoyed it.
Making this time. So, all right. Bye, guys.
Talk to y'all later.
Who are our potential consumers? Well, it's anybody wanting to maximize their health span, be healthier for longer. It's people wanting to maximize their lifespan. People wanting to maximize bedroom performance. People wanting to look younger for longer. A lot of the girls say it's better than Botox because it keeps the skin cells younger. Athletes, because normally they'd peak out fitness-wise at about 25. It's everyone. And everyone suffers telomere shortening. And in addition to providing telomere support, isogenesis was developed to improve overall health and well-being. Plenty of people are experiencing great results. Enhanced energy and stamina, improved quality of sleep, faster recovery after exercise, better memory, better focus, concentration, and understanding. I mean, it's fairly simple. We take 2 capsules twice per day. It's 120 capsules in a container, so one container lasts 1 month. Works out less than $100 a month. Works out less than the cost of a cup of coffee per day. Volume discounts are available. And Isagenix have a 30-day money-back guarantee. So just try the products. You've got nothing to lose. If you want to find out more information or order the product, get back with the person who shared this information with you. Thank you.